what's happening here. And so with the regulation of gene expression, there are basically two areas where we really see this play out very clearly in eukaryotic life forms. And they're both in multicellular eukaryotic life forms. One is in creating the division of labor. Like to where you can take two cells that are genetically identical, but have one become a liver cell and one become a neuron. And you do this with changing which genes are expressed. Another place we see this is in a cell's response to external signals. That when cells are involved in communication, which we talked about in the last chapter, right? So they receive a signal or bind the ligand, step 1. Step 2 is to transduce that signal. Step 3 is respond. Most often that response is going to be a change in the gene expression. Okay, start making something that they weren't making before, stop making something they were making before, or make more or make less. Okay, oftentimes that's what the response is going to be, altering the gene expression. So again, see it pretty much two places in eukaryotes. Division of labor, basically cementing this cell to become a certain cell, and changing the expression of a gene in response to some signal. Now, which of those do you think is a more permanent change? To facilitate the division of labor or to respond to some external signal? To division of labor. And so what's interesting is you should be anticipating there's probably at least two ways in which we can regulate gene expression then. We can permanently shut down a certain gene to where it's never expressed, or we can change the rate of the expression. Okay, but before we talk about eukaryotes, we need to talk about prokaryotes first, because in many ways their regulation of gene expression is a lot simpler than eukaryotes. And so we have this question here: how do operons provide a mechanism for altering gene expression? How do operons provide a mechanism for altering gene expression? I will tell you this: in prokaryotes, the only way to alter the gene expression is through these operons. Because you know prokaryotes lack something that eukaryotes have that's part of the name. And what is that? What does prokaryos mean? First nucleus. And eukaryos means true nucleus. Okay? Prokaryotes lack a nucleus. And so when we learned about gene expression, we learned transcription happens in the nucleus, right? Where the DNA is written into RNA, RNA polymerase writes an RNA copy of that DNA, then it is modified, escorted, if you will, that's a good term, escorted out of the nucleus where it gets translated. There's no nucleus in prokaryotic cells, so the transcription and the translation happen in the same place at the same time, okay? And so it limits your ability to regulate that process, but operons help us do that. So how do operons provide a mechanism for altering gene expression? So here is the most well-known of all of our operons. And so when you look at this, what do you see? Okay, we've got polymerase, so we know we're talking about, and it's RNA polymerase specifically, so we're talking about transcription, meaning now we're talking about a gene. And we talked a little bit about promoters before, and right, we talked about the Tata box, yeah? And how in eukaryotes that basically is a signal of a coding gene. In prokaryotes, the promoter's different. We call it the PRIBNOW sequence. But anyways, we've got this promoter that tells RNA polymerase where to bind, where to start writing. Yes? Okay? And so we're looking at this, and here we have a promoter, and then we have 1, 2, 3 blocks. You see that? So we've got our promoter, and we've got 1, 2, 3 blocks. And each of these blocks is a separate gene. Each of these 3 blocks is a separate gene, and it is a separate gene that is translated into a separate protein. We've seen this before, beta-galactosidase. Anyone remember what that enzyme does? Yeah. It cuts lactose into galactose and glucose. As we talked about this in lactose intolerance, if you don't have that enzyme or the bacteria in your gut don't make that enzyme, you don't break lactose. And so this is required to break lactose into its individual monomers, monosaccharides. Permease is required to get lactose into the cell. And then transacetylase is required to basically prevent the metabolism of other sugars in the presence of lactose. So all three of these genes are associated with metabolizing lactose as a sugar. And that's why we call them LAC-Z, LAC-Y, and LAC-A. You're like, wow, that's horrible. Why couldn't they have gone in alphabetical order? I don't know. They didn't even go in reverse alphabetical order because it's like LAC-Z, LAC-Y, LAC-X, but it goes to LAC-A. But so what happens, if we have three genes with a single promoter, they are transcribed as a single unit, right? But when they're translated, they end up generating different polypeptides. Because at the end of this, you'll have a stop codon. Okay? And at the end of this, you'd have another stop codon. But so at the end of this, you have a stop, and then you have a start codon somewhere here, and then a stop and a start codon somewhere here. So they're transcribed as a single unit, but when they get translated, they become three separate polypeptides. But all three polypeptides are functioning again in the metabolism of lactose. Okay? Make sense? Now, there's something else on this diagram that we haven't talked about before. Do you see anything on this sequence of DNA that we haven't talked about before? Well, there's LAC-L, right? And then lots of LACs. We've talked about promoters. Have we ever talked about an operator? That's new. And look, operator starts with the same four letters as operon. Coincidence? I don't think so. And so this operator is regulating the expression of this region. And so you see here there's a separate gene called the LAC-L gene, and its product is the LAC repressor. You see that? The LAC repressor, and this protein goes and sits down right there at the operator. And so you've got basically a big old block sitting right here. So as RNA polymerase comes in, it can't get past that block. That make sense? Does that make sense? It's like you've got one of those big concrete barricades preventing you from taking this road that would be an awesome shortcut and would take way less time. But for some reason, nobody wants you to take that road because it cuts through people's houses. So selfish, right? Oh gosh, we have one here right on Placerita Canyon Road. I wasn't even thinking about this. I was thinking over by my house though. But it's like, we don't want trucks taking this road anymore. We'll put a gate up. You can't even use it. Have you ever had an Uber come and pick you up from here? And then they pair you with somebody that's over on Sierra Highway because they think that they can come up Placerita Canyon? And you're like, no, it's going to take them 15 minutes to get here. And they're like, no, they're 2 minutes away. You're like, nope, cancel. And then you do it again to get one that's actually on Bouquet. Y'all don't take Uber anywhere? You're like, no, I have my own car, thank you very much. So do I. But it's still, anyway, sorry. So this block goes and sits there and prevents the transcription of these genes. Because the bacteria does not need to transcribe these unless lactose is present. When lactose is present, now it needs to metabolize lactose. If lactose isn't present, it has no need to metabolize lactose. That make sense? And so you're like, cool. The operator's just sitting there preventing transcription. I get it. How do we get this operator off of there if we want to transcribe it? It's a wonderful question. So, now, prokaryotic cells are designed in very interesting ways. When you have a metabolic pathway, a biochemical pathway that requires several proteins, those proteins tend to be located like they are here, where all three proteins are transcribed as a single unit like this, where all three proteins are right next to each other in the genome. And so, also are often adjacent to one another. And so what this means is if you stop the transcription of one of them, you stop the transcription of all of them. Okay? Which makes sense because they're all involved in the same biochemical process. These regulatory proteins literally bind onto the DNA at those sequences to prevent transcription. So the promoter is one of these regulatory sequences, and the operator is the other. And all of these genes of an operon are transcribed into a single mRNA. We already mentioned this. And so then we've got an operator, another regulatory element, that allows things to bind to it to shut down, or sometimes we'll see this with another operator, accelerate the transcription of those genes. Okay? Any questions? This is just in general. We're going to look at this more specifically because I know this is not enough, right? It's not enough. I didn't feed you enough yet, but I soon will. And I don't know if you get this sense after you leave this class, but I really don't like people to come over and to leave hungry. Like, I like you to leave full. It's a great analogy for what happens. So they have a rule when, like, you're having people over to do half a pound of meat per person. But I eat more than half a pound of meat, and I expect other people to as well. So I like to go 1.5 pounds of meat per person. And then you send some home with people. You know, it's like you go to a birthday party and you go home with a little party favor. I expect you to leave my house with like some food to take home with you too. And then that way you don't ever want to come back because you left with food. You're like, why do I need to go back? Because I don't want you to come over to my house multiple times for dinner, right? It's like one time you came over for dinner, enjoy the rest of your life, you know? So what are the components of the lac operon? What are the components of the lac operon? Well, many of these we've already talked about. So here, wow, that color looks terrible. Looks so good on here, but on here it looks like a highlighter. Cool. You like highlighters? I don't like it. Okay, so here we've got the repressor. Again, this is a separate gene. This is a gene that's not part of the operon that transcribes this repressor, but this pressor goes and sits on that operator to block the transcription. So RNA polymerase, even if it binds here at the promoter, it can't write anything and just falls off. And then 3, that's where we would get the rest of our gene. So here, now you see it here, we've got an inactive repressor. So there's something here that is binding to this repressor and getting it to basically fall off of this. Now notice what that repressor, or what inactivates the repressor. Do you see this? Can you read that? Allolactose, which is a form of lactose. So when lactose is present, when that sugar is present, look at this. When the sugar is present, it binds to the repressor better than that repressor binds to the DNA. And so now it allows you to transcribe the genes necessary to metabolize what bound this repressor. So the key that unlocks this lock is what this is going to actually eat. So it's like the key is a donut, and you unlock the lock and then you eat the donut. It's so beautiful. It's like, how do I eat this donut? Right now there's something on my face, but the donut is the key to unlock the thing that's on your face. You're like, why is there something on my face that's locked? To keep you from eating donuts. It's all a conspiracy. The gluten-free people that don't want you to eat donuts want everybody to be miserable, right? Oh, I got to tell my wife she's got to listen to this one. I was like, this is a good one. This is a good one. All right. Lac operon contains 3 different genes, LAC-Z, LAC-Y, and LAC-A. We've already mentioned those. All 3 of those are necessary for the metabolism of lactose. Okay, all 3 of those are necessary for the metabolism of lactose. We call this a negative regulated operon, where the default position is off. So the default position of this operon is off. The default position, that repressor sitting there, you're not transcribing it. And so it only gets turned on when lactose is present. Okay? When lactose is present, it gets turned on, and then you can metabolize that lactose. Now that repressor is coded by a separate gene called LAC-L, separate from the operon. Another name for this is what's called an inducible operon, one in which you can turn it on. Okay? And then we have repressible operons that can be turned off, but the default position is on. Okay? But this one, the default position is off. And you're like, why would the default position be off? I'm glad you asked. Why would the default position be off? Why would the default be don't transcribe the proteins necessary to metabolize lactose?
Have too much and no lactose for it to do anything with?
Yeah, lactose is not a really, why is the word popular coming to my mind? I mean, it is a really popular, sugar because it's in milk and ice cream and cheese, and, those are wonderful things to eat. It's not a common sugar. There are other sugars that are far more common to eat, and I don't know if you knew this or not, but our bodies are not really designed to consume dairy for our entire life, which is why there's a period of nursing when you're young, and there's lactose in human breast milk, and there's lactose in basically all mammal milk, and then it's like you get weaned and you start eating real food, and so our bodies aren't really designed to actually continue to eat lactose throughout our life. And what's interesting is the wild type condition is probably to not be able to consume lactose. And so we as humans have developed the ability to continue to consume dairy throughout our lives, probably by being genetically modified or modifying our gut biome, by viruses, Anyways, so that way we can continue to eat cheese throughout our life. If you're lactose intolerant, I'm really sorry. Yeah. You're welcome. Yeah. You're welcome. If you come over to my house for a dinner, I will still make some, but I'll provide Lactaid for you. All right. So if you had a mutation in this lac operon, how would it change the expression? Let's just say for a minute, okay? Let's play in hypotheticals. Let's say you had a mutation in the lac Z gene, and it was a missense mutation. Okay? So you're like, missense mutation, I know what that means. It means I switch the one amino acid out for another, and then you're like, the effect of that on that protein is going to be determined by what? The chemistry of the amino acids, right? If we switch the amino acid out for another amino acid, but they have similar chemistries, that protein's going to still function. But if we have a missense mutation and switch it out for a chemically different amino acid, now we're not going to be able to make beta-galactosidase. We're not going to be able to metabolize lactose. But what if your mutation is in the LAC-L gene? What if we have a mutation in the LAC-L gene that prevents the product from forming? What does the LAC-L gene do again? Somebody remind me. I've forgotten. Yeah, Allison. It has the code for the repressor for the operon, right? So if you have a mutation in that gene, you don't make that repressor, guess what? Now instead of the default position being off, now the default position is on. And this cell is constantly making proteins to metabolize a sugar that may never show up. You're like, wow, that's so wasteful. It is. And that bacterium probably is going to invest a lot of energy in doing something that is useless and will be outcompeted by bacteria without a mutation. Who knew? Okay, so the next question we're going to address is this. Well, when is this on? If it's an inducible operon and its default position is off, when will this be on? Okay, and we're going to take ourselves a little lecture break. You already know one answer to this question, and that is when lactose is present. But what I want you to do, take some time, work with those around you, and I want you to propose some other conditions that probably need to be met in order for this operon to actually be on, because I will tell you, something has to be true in addition to there being lactose. Okay? When lactose is there, it'll turn it on, but not really. It's kind of like a dimmer switch. Y'all know about dimmer switches? Where it's like you can turn it all the way on or you can turn it just a little bit on. Yeah, they oftentimes put them like on the light fixtures above dining room tables because they think some people like to eat in the almost dark, right? You're like, oh, it's totally fun. It's like, then just light a candle. Or go eat in a bathroom with no windows and it's completely dark. You know, there are restaurants that do that, right? That it's completely black inside and they walk you to your table because when you can't see, your other senses are heightened. So you taste better and you smell better when you can't see. Yeah. That's just what I'm saying. That's what I'm saying. I'm way too paranoid for that. I don't like to sit in a regular restaurant with my back to the door. I always sit place where I can see all the exits. I'm not going to go into a restaurant where you can't see it all. Gosh, silly. Okay, sorry. So in addition to lactose being present, what do you think has to also be true for that switch to be turned all the way on? So not only are you transcribing this block of genes, but you are transcribing at a high rate. Okay? Take a few minutes, take, I don't know, 90 seconds, talk about this with those around you. 90 seconds, starting now.
What else has to be present for
It's a lactose, a combination of lactose.
Depends on what he's going to do.
He did have glucose, you just have lactose.
Yeah, take that powders off, like with what you get with the final
But that's the thing, because
So lactose is present, but you
does
don't
this have
have
to do with glucose?
I mean, the transcription, whatever. Yeah. Yeah. You want that
ask you a question?
I wasn't here. I stepped out first.
Sorry it took so long. I spilled water and made a huge mess. I had to clean it up. Sorry. I was gone for 2 minutes instead of 90 seconds. What do you think? I know. It's inexcusable. But I'll let you know I only cleaned up about 75% of my mess. Because I want to, I want people to know I was there. All right. So what else do you think needs to be true in order for this to be all the way on? Rather than just a little bit on. No thoughts? Yeah, Davies.
if you think of lactose, it's made up of probably three monomers, I would say.
Two. Yeah.
So galactose and glucose.
Yes.
And so if you think of glucose being absent in that stage, there will only be galactose and it will fail to, like the operator will fail to attach itself to
Okay.
the
So like if you have something other than other.
lactose.
Okay. Yeah, Autumn.
Is there an enzyme that has to be there?
I mean, sort of indirectly. So you also have to be deficient in easier sugars to metabolize. Because even though lactose is present, there are easier sugars to metabolize, right? Glucose it's already a monosaccharide. It's half of lactose. And so if glucose is present, even if lactose is present, you're still not going to really transcribe this at its maximum rate. So here we already talked about this. Allolactose, a form of lactose, basically binding to the repressor. And so we have another thing here. You see this? CAMP stands for cyclic AMP. And what it does is it increases the rate of the transcription of this operon. Okay, this molecule cyclic AMP and it's a derivative of AMP, which is adenosine monophosphate. It's just like ATP, but instead of 3 phosphate groups, it has how many? Just 1. Okay? And cyclic AMP, what this does is it basically increases the rate of transcription. And look when cyclic AMP the numbers of cyclic AMP are high. When lactose is present and glucose is low or absent. And so let me show you. Oh, yeah. And so here's similar scenario. Lactose is present, but glucose is present as well. The repressor's not there binding because lactose has bound that repressor. You see, the repressor's not there at the operator, but without cyclic AMP, very little of this RNA polymerase binds and actually transcribes that operon. Yeah.
So is that the reason that young mammals process milk better, because that's all that they're taking in?
It's the only sugar you're really taking in is lactose, and that's probably part of it, because a lot of it has to do with your gut bacteria and your ability to process that. The lactose content in human breast milk is much lower than in cow milk as well. So there are a lot of things contributing to it. But yeah, I mean, it is basically the only sugar present when you are on your breast milk diet at the beginning. Yeah.
So if you ate something with glucose, like raw sugar and lactose, your body would metabolize the glucose first?
Yeah, well, a lot of the, yes. I mean, the bacteria. What would it do with the lactose?
Would it save it until the glucose wasn't there?
Maybe. Yeah, maybe. Although you'd still have those enzymes that would be breaking it down. That's a good question. That's a good question to know if there are people that are only, because what you're getting at is, could it be possible that people are only really lactose intolerant if they consume lactose at the same time as consuming other sugars? That's intriguing. That's worth looking into. We need somebody who's lactose intolerant that's willing to allow us to experiment with them. You volunteers tribute. I mean, during our final exam would be the best time to do this experiment as well. Actually, about an hour before the final exam starts would be the best time to conduct this experiment.
Just so you're feeling real good.
Yeah. Yeah. We want to make sure you're at your peak performance during the final exam. All right. So the lac operon's only active when lactose is present and glucose is in low numbers, or is it not present at all? And again, we've got this cap protein activated by cyclic AMP when glucose is absent that really allows RNA polymerase to transcribe enough of this operon that you can reasonably metabolize lactose. And so we've already talked about this a little bit, but why wouldn't the lac operon be active when glucose is present? Yeah.
Because then you'd be wasting like unnecessary energy.
Yeah, I mean, you'd be wasting energy to break down a sugar when you have plenty of glucose that's already present and much easier to metabolize. So now this one is an interesting question. So this operon really has 4 genes. The 3 genes that are actually part of the operon, plus that 4th gene that codes for the repressor, right? That turns it off. And so the question you have is this: how do you develop this operon if you don't already have it? There's only one way to do that, and that's if it comes over as the entire unit through some kind of horizontal gene transfer, which we know can happen in bacteria because bacteria will transfer information through conjugation from one bacterium to another. Bacteria can transfer information horizontally through viruses, call that transduction, or bacteria can just pull on foreign DNA. Okay? Does it make sense? Any questions? But you have to get it as a unit, otherwise it's not functional. One of those, lac Z is not enough to metabolize lactose. Lac Y is not enough to metabolize lactose. Lac A is not enough. You need all 3 of them to metabolize lactose. And you need lac L to regulate the transcription of that operon. Okay. Is that cool? Does it make sense why we call it an inducible operon? Default position is off, but it can be turned on. Okay? Here's an operon that the default position is on, but it can be turned off. Okay? This is called the trip operon or the TRP operon. And again, this is what we call a repressible operon. The default position is on, but it can be turned off. All right, so here is the repressor in the absence of tryptophan. So tryptophan is one of the amino acids. And if these bacteria are living in an environment where there is not tryptophan present, they are making tryptophan. Okay? And in order to make tryptophan, they need 1, 2, 3, 4, 5 separate genes to make that amino acid tryptophan. You're like, why would they need to make tryptophan? It's an excellent question. Why would they need to make tryptophan? It's one of the amino acids. And what do we build out of amino acids? Proteins. Why do they need tryptophan? Because some proteins require tryptophan. Does that make sense? Okay. So if they're in an environment where they're not picking that up in their diet, then they have to build it. And so the default position of this operon is on. The default position is on, and this bacterium is making tryptophan. But it's a repressible operator, or a repressible operon. Here's the operator. So the default position is this is inactive. You see that? It's inactive, so it can't bind here. It's already there. It's being made by a separate gene, just like the repressor for the lac operon was made by a separate gene, lac-L. But now this default position is inactive. What do you think activates this? Tryptophan. And so the very thing that this is making, okay, if it's not using it right away to build proteins, then it would turn this off. And if they start taking this in from their environment, so here's the repressor in the presence of tryptophan. Now they're taking in tryptophan. They're also making it. There's a ton of it. Some of it binds to the inactive repressor, turns that repressor on, and then that repressor can go and sit right on that operator and prevent the transcription of those genes. Okay? So again, lac operon, it's an inducible operon. Default position is off, but it can be turned on. This right here is a repressible operator. Default position is on, but it can be turned off. And that's, again, even with the lac operon, what deactivated the repressor was what? What deactivated the repressor? What was the key that unlocked the mass preventing you from eating the donut? The donut, right? It was lactose. And here, it's the same thing. What they're making actually acts to now put the lock on to prevent them from making it. Yeah, David.
So if it needs to get turned on again, how does it?
Well, once the level of tryptophan drops to a certain point, then it'll get turned back on.
So like that tryptophan is gonna get kicked out, or?
Yeah, when the bacterium needs it to actually build a protein, yes.
Okay.
Yep.
So does that say that tryptophan is more important than, or more readily used than lactose?
Yes. Yep. Oh yeah. Yep. Because lactose you're only going to use if you have no other sugar option, right? Or at least no other simple sugar option. But tryptophan you're going to use all the time whenever you're making proteins. And so to summarize this, the TRP or the TRP operon is a repressible operon. And when it's active, it allows the E. coli to synthesize tryptophan. It's got 5 structural genes necessary to make a single amino acid, which is fascinating to me. That to make a single amino acid, one of 20 that are required to make proteins, requires 5 proteins to make one block. If you want to use the analogy of like Legos, where these amino acids are Legos that you're using to build, in order to make one Lego, and you're like, this isn't even a well-used or often used Lego. This is like one of those weird, awkward shaped Legos that aren't really valuable unless you're making something weird. But in order to make it, you need 5 different machines built out of Legos to make that one obscure Lego. Like, this is really interesting. They're really interesting. How do things like this just happen? And then, so this tryptophan deactivates the operon by activating the repressor. And so in this one, the repressor is being synthesized, but it's being synthesized in inactive form. And so the tryptophan activates the repressor, and then that repressor can then block the transcription. And so you have this question. I think this is a fun one to talk about. We'll take another lecture break, and I want you to work on addressing this question with those around you. Which of these operons, the lac or the trip, is more complex? Which of them requires higher levels of control? All right, take a little bit of time, see if you can come up with an answer and a justification for your answer.
And this one's rarely going.
It's all active like one con. It's like last resort.
Yeah.
Yeah, because not only like does it have to regulate plant, but then you just shut it off. So like months to making it right. So that it's like
This one.
if you're using it, you're making it more, that means it's shutting it off.
You make it
I think the tryptophan one.
kind of curious.
But also, you know, it wastes energy.
So when you're making something,
Because like it's like an issue of like it also means like they also like have to make it on their own as well. They like regulate that as well. Whereas lactose is just like another option for sugar that they can use to just break down for energy. Whereas like this is something they need to build stuff and when they don't have it they have to synthesize it. So they have to regulate that. But like if it's in the environment then they like down regulate it, like it's synthesizing it, up regulate whatever. Or, yeah, down regulating whatever. But
Okay, what do we think? Which one's more complex? Which one has higher levels of control? So what's interesting is the lac operon is not on even when the repressor is not active, right? So when lac binds to the repressor and inactivates it, it's still not fully on unless you have a second protein called the cap protein helping RNA polymerase bind. So with the lac operon, it's both the operator regulating the transcription and a second protein called the cap protein regulating the transcription. So as far as levels of control are concerned, the lac operon is more sophisticated. So it's not just a matter of getting the repressor to not bind. It's also you have to get that cap protein to recruit and encourage RNA polymerase. And so then you're like, okay, so it's more complex. There's a higher level of control. And the question is why? Why is there a higher level of control? And there are two decent answers to that question. Not great because we're still trying to figure this out. One is you don't want to metabolize something when there's something easier to metabolize. It's wasteful. And then second is once you start metabolizing something, you have the potential to build up other metabolic waste products, okay? So when you metabolize lactose, you break it into galactose and glucose, okay? And now you have two different monosaccharides you have to deal with. Glucose, very easy to deal with. Galactose, easy to deal with, but still needs to be modified a little bit to be able to be taken into glycolysis like glucose is. Whereas our other operon, our trip operon, it's like you need tryptophan. So if it's not present in your diet, you need to make it. Okay, there's really no other choice. Now, if you have it, you don't want to be wasteful and keep making it, but you're not dealing with the potential. You're only really dealing with waste, like energetic waste. You're not also dealing with a potential buildup of metabolic waste products that can be harmful. All right? Does that make any sense? It makes sense in my head, but it's Monday of the very last week before final exams. Mondays are always a little bit sketchy, And the Monday of the week right before finals exam, especially sketchy. Yeah. My brain's a little squishy. I don't know about yours. So, do you ever feel like sometimes, you know those stress balls that you squeeze to like alleviate the stress? Do you ever feel like life is just kind of treating you like a stress ball? Yeah. All right. Well, we're in a good position right now. We're in a good position right now to not go on to our next one. And then on Wednesday, we'll start in and talking about eukaryotic gene expression. Right now.